Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show, bringing you another really interesting guest today involved in creating a better tomorrow for many people out there. Today, we have the honor of being joined by Dr. Scott O'Neill, who is a professor and director of the World Mosquito Program, located at Monash University, which is a large international research collaboration, formerly known as Eliminate Dengue, which has focused on developing the Wolbachia bacteria as a really novel method to block the transmission of dengue fever and other mosquito-transmitted diseases, such as Zika, Chikungunya. And the nonprofit program is currently conducting field trials of the so-called Wolbachia method in multiple countries around the world in areas where these diseases are endemic, ultimately working closely with communities and local health organizations and governments to implement this self-sustaining method through controlled releases of mosquitoes. Dr. O'Neill spent his academic career at various locations, including University of Illinois, Yale, the University of Queensland, and Monash, where he was previously the Dean of the Faculty of Science. Dr. O'Neill is internationally recognized for his various contributions to the field of insect symbiosis and the way that these intimate bacterial associations with invertebrates can generate really interesting effects on reproductive physiology, developmental biology, ecology of the insects they infect, and his work has made major contributions to ultimately understanding how these insect symbionts ultimately affect and the consequences for their infective hosts. We're honored to have him with us today. A lot of really interesting themes to get into, but Dr. Scott O'Neill, thank you so much for taking the time to come on the show today.
You're welcome. Pleasure to be here.
It's great having you, Scott. I'd love to start off as we typically do, just by giving you the floor for a little bit to talk a bit about your background story. I looked back at your dissertation, symbiont-induced sterility in insects. This is back in 1989. So you've been very focused on this theme for a while, and you're publishing. It wasn't just about mosquitoes at the time. You were publishing on parasites in Drosophila. You were publishing on this really interesting thing called the confused flower beetle, which causes all sorts of problems for grain storage. Talk about how you got started in the whole area of the symbiosis story.
So I had, I was doing a PhD in Australia at the University of Queensland, and my professor who was supervising me was an evolutionary biologist, Hugh Patterson, South African scientist, and originally, and he was very interested in Wolbachia and the possible contributions it may make to evolution of insects, and he had a background in mosquitoes. So he sort of introduced me to the whole notion of Wolbachia and this intracellular bacteria that did all these amazing things in insects, as we found out over the years. But that was my first introduction to it back then. It's funny that you pulled up my thesis title that you went and looked at. It seems like 5 ages ago to me now that I was a student doing that work. And I got very interested in Wolbachia and particularly how it could be applied to insect-transmitted diseases. And so I got more and more into mosquitoes, less into flower beetles and other types of insect pests, and have been focused on it ever since. So it's been a lifelong obsession for me, I guess you could say, as I'm getting towards the end of my career now, towards it. I've been working on it for many, many years.
Got it. And talk a little bit about, I mean, it was Peter Singer from the World Health Organization that actually brought up the topic a few months ago when we had him on the show. It's a really interesting bacteria, and I was struck to find that it's really found throughout many arthropod species, and it does a fairly from affecting the mating patterns to and so this theme of cytoplasmic incompatibility, which you published on a lot, into some other really unique biology. I had to look up what philatoky was, and sort of this parthenogenic mechanism of getting rid of males entirely and creating only females. Introduce us to Wolbachia a bit, and the fact that it is so prevalent was kind of like, why are most of these insects around nowadays anyway, if it's everywhere? But talk a little bit about it, if you would.
Yeah, so Wolbachia is incredibly interesting bacteria. Firstly, it's intracellular. So, it lives inside the cells of host insects that it lives in. And what's unusual about it compared to most infectious organisms or most organisms that are parasitic or pathogenic is that it's not infectious. So it doesn't transfer between, like COVID does, from one individual to the next. We would call that horizontal transmission. You get virus introduced into your body and then from the person next to you. Well, Wolbachia doesn't transmit that way. It's not able to transmit that way. So if you can't transmit that way, what other tricks could you have to get into other hosts? Because you can't stay in one host all the time because that host will die, and so you need to propagate into new hosts, and then to be able to maintain yourself in that host population. And so Wolbachia has this amazing bag of tricks that it uses to ensure that it's able to spread into an insect population and maintain itself. So things like cytoplasmic incompatibility, which is a little complicated to explain, but in the simplest form, what it is that there's, if you have this bacteria and you're a male and you mate with a female that doesn't have it, then all of her eggs die when they're laid. And that gives an advantage to the other females in the population that have Wolbachia. And that's because only females transmit Wolbachia. It's packed into the eggs of the insect. It's not in the sperm. So males are a dead end, but females are not. And so this is a way of indirectly benefiting the females that have Wolbachia and helping it to invade the insect population and maintain itself. Another example is sex ratio distortion, or parthenogenesis, as you mentioned. It will skew individuals to be able to reproduce without males and convert males, genetic males, we think of ourselves as mammals as an XY is a male and an XX is a female. It'll take an insect the equivalent of the XY genetic male and turn it into a female. And then that female will then lay eggs that contain Wolbachia. So it has all these methods and a number of other ones that are able to, hijack the reproductive biology of its host to ensure that it gets transmitted into the population and so maintains itself. So in that way, it's been able to find pathways to ensure its survival and success that infectious agents like viruses or whatever don't need because they have a direct way. But this parasite uses indirect methods to maintain itself in insect populations. So it's very unusual. It's very successful at doing this, and we now think that possibly more than 50% of all insect species on the planet have this infection. Because of these mechanisms it has, it makes it so successful to be able to invade host species.
Got it.
And I should say that when you think about how many insect species there are on the planet, so take a minute to think about that, how many insects are on the planet. Well, we've got no idea. There's a lot. Many millions of insect species, and maybe 50% of them have this bacteria naturally. It's probably one of the most successful bacterial parasites on the planet. when you think about how many insect host species it's able to infect and establish itself within their populations.
Absolutely, the miracles of of evolution. But yeah, no, it is a
Yeah, it's
fascinating
a miracle of
array.
biology. It's
Yeah,
it's incredibly unusual, incredibly successful, and so I started out with an interest in just understanding the biology of the Wolbachia, and we did the early work showing that it was very widespread in insects, and it was associated with these different phenotypes, etc., in the insect hosts. But then, my real interest was about applied biology and how could we use Wolbachia in a very practical sense
got it.
to have benefit for human populations, essentially.
And when we look at the sort of the array of nasty things that mosquitoes spread, and obviously, I mentioned those in the intro, whether it's dengue or Zika, malaria, of course, or West Nile. We touched on some of these when we had Peter Hotez on a few months ago, from purely the perspective of hey, these are neglected tropical diseases that we got to do a lot of stuff in terms of this or the vaccine work. Let's talk a little bit about mosquitoes in general and some of these pathogens, because I guess I guess in our neighborhood here, I guess had some run-in with Zika a few years ago. I guess most people on the East Coast of the United States maybe don't run into malaria, dengue, obviously, not so much either. But this is a major issue. And just looking at the numbers, close to 400 million people infected every year, and a significant amount of mortality and morbidity associated with this one. Take just a little into sort of how you see the landscape of mosquito-borne illness in 2023.
Yeah. So there's two main groups of, if you like, of pathogens that mosquitoes transmit. One group are the parasites. You mentioned malaria, which we've known about for many years, which is a huge, huge problem. It's probably the biggest problem, or is the biggest health problem associated with mosquitoes. But then there's all these other, a bit more obscure virus diseases. And, you mentioned some of them, Zika, and when we had the big outbreak in 2016, and discovering that Zika was causing abnormalities in fetuses and was having these birth defects. It was horrific for people. People, say, on the east coast of the US, where they didn't have to directly deal with it, but were seriously concerned about, should you go on holidays to Puerto Rico or Brazil or wherever you might be going? Well, sure, but imagine if you're living in those locations. And so what we saw is that, there was people were avoiding getting pregnant and having children during this period because of so much fear. And so Zika, a very recent one. Dengue is the big one. 40% of the world's population at risk of dengue, massive outbreaks, having a very bad year with dengue in Southeast Asia this year. And part of the fear with diseases like dengue is similar to the fear that with COVID. When COVID arrived into town, do you remember the images of them building a hospital in China in 48 hours? And then the ship that came up to New York, the hospital ship. The fear at that time, which no one was really talking about, was that the health system would collapse, that they would be so overrun with patients that needed to be addressed that, there wasn't going to be the capacity. And so there was a huge fear about stopping transmission and not letting the health system collapse. Well, it's the same thing with dengue. It's just that the mosquito that transmits dengue only lives in the tropics or can extend up into the subtropics. So currently, if you're living in New York, it's not an issue for you. But if you're living anywhere in the subtropics or tropics anywhere in the world, you're at risk of getting this disease, and it can kill you, and there's no treatment for it. And if you live, I'm currently living in France, but prior to that, I spent the last 3 years living in Ho Chi Minh City in Vietnam, where there's a lot of dengue. And here I am, I'm like a mosquito expert, apparently. And I couldn't control the mosquitoes around my house in Vietnam. It's impossible not to get bitten. There's just so many there. And so for people that live in the tropics, there's a lot of fear associated with picking up a disease like dengue, or your children picking it up, and then the possibility that they might die of it, and you can't really protect yourself. And then there are other diseases that are very famous, like yellow fever. The stories of yellow fever and the Panama Canal,
Yep.
and how so many people were killed from yellow fever transmitted by the mosquitoes. And what's particularly interesting, and it relates to the work we do, is that those diseases I just wheeled off, Zika, dengue, yellow fever, they're all transmitted by the same type of mosquito. This mosquito we call Aedes aegypti is the major vector. This one mosquito is transmitting all of these viruses to us. And so our focus has been primarily on that one mosquito to be able to prevent it from transmitting all of these different pathogens, which is that's such a problem for human populations, particularly in developing countries, particularly where there's no good social safety net for people, and health systems are fragile, and the consequences of the diseases can be huge for people. Some people can't afford to lose a couple of months off work. Some people can't afford to have a disabled baby that they then have to care for a number of years before it dies, without any assistance to do that when they're living on the poverty line themselves. And so the social and economic consequences of these diseases are enormous, not just the mortality figures. Actually, the mortality figures are not that important. It's much more about the social and economic costs of having this disease burden persist in populations, particularly in poor countries.
So now, after laying that groundwork, and again, for everybody listening and watching the show, we'll put the link to the World Mosquito Program out there, and you can see sort of the timeline that Scott's talking about here. And I like the one part where you see the discovery of Wolbachia, 1924. You start working on things in 1980, doing a lot of amazing research, and again, we can find Dr. O'Neill's Google Scholar site out there, sort of the sheer amount of papers that have been put out on this topic. The organization is formed, and you get some really important funders like Bill and Melinda Gates Foundation involved. Before we get into sort of the dynamics of exactly what you're doing with the eggs and the release and all that, talk a little bit about the history of the evolution of the World Mosquito Program alongside, your own intellectual work and in the whole area of insect symbiosis, ecology, and so forth. Give us a little of that history, if you would.
Yeah, maybe go back before the Gates Foundation got involved. And I'm an Australian scientist, trained in Australia, and I was fortunate enough after my PhD to get a position working in the University of Illinois, and being supervised by Hugh Robertson, and that was an incredibly eye-opening experience for me. What a University of Illinois is! For me, coming from Australia, it was like to be in a university that had a number of Nobel laureates on campus, that just had, it had two entomology departments there, not one, but two. It was an incredible experience. And then I was fortunate enough to get a job offer at Yale University, and I went there as a junior faculty member. And for a number of years, maybe 10 years, 8 years or something, I was grinding it out like a junior academic would, living. I don't know, I've always thought of myself as a professional beggar. And so, you spend your life trying to get money to get a patron to support your ideas and your work. And, you just write grant proposals one after another. And I was fortunate enough to receive, National Institutes of Health funding in the US, and the NIH supported my early work for quite a while. And during that period, we had ideas about what we wanted to do, about how we could use this thing called Wolbachia for disease control, but we never had enough money really to do anything profound with it. We kept on pushing it. trying ideas. But what we were finding that most of our things we were suggesting and wanting to do were failing. I had like many years of great ideas that went nowhere with experimental work that we couldn't pull off technically. And then back in 2003, the Gates Foundation ran a competition called the Grand Challenges in Global Health. And at that point, I just returned to Australia and I'd taken up a position to head up a biology department at the university where I received my PhD. And this call for proposals came out, and I couldn't really help myself. I'd sort of got to the point where I was thinking, I can't make this work. I should do something else. And I can't burn any more PhD students' careers on these ideas. And then this Gates Foundation proposal came out, and it was right in my zone. It was about mosquito-transmitted diseases, novel ideas to find solutions for the problem. And so I dusted off, the old proposals that I'd been writing and put this letter of intent in, and not thinking much more about it, being the professional beggar that I am, you're churning out requests continuously for money. And lo and behold, they ran this global competition, and I think they had about 1,800 ideas came in, and they funded 44 or something. And we were one of those ideas, and we got like $6.7 million US, and my grant sizes for me before that were 250,000 or something. And so at that point in time, 6.7 million was a large amount of money. And so at that time, I was just ecstatic that we could land that money to finally, really try this out. And at the same time, this sort of sense of ecstasy, getting a big drop in my beggar's cup, but then the sort of clenching of needing to have to actually deliver on what I said I'd do, and knowing that I'd failed for the 10 previous years, and in being able to deliver this. And so we pitched and we were able to sell it, and now we had to deliver it. And after many years of failure, at that point, just magically, it seemed, stuff started working. And it's been working ever since, actually. And after, the first half of my career, nothing really working, and now moved to a point where everything started working. So it was an interesting inflection for us. Yeah. But certainly that pulse of money from the Gates Foundation really allowed rapid progress for our ideas, and opened the door for us to really try out whether this could work or not, what we wanted to do. Could we have an impact, a positive impact on disease?
And how did the evolution of the protein / waste container model, which I can only describe as what looks like a large sort of milkshake container, for the controlled releases of these eggs, how did that model come into play? Obviously, because when we read about, we think about this, or we sort of read the headlines of the articles all about the program, and it sort of sounds like there's this, it's just about releasing all these mosquitoes. No, there's a little process here, very elegant, and there's this cool article you wrote, Trash to Treasure, specifically on this particular model. Talk a little bit about that, if you would.
Yeah, well, maybe I'll back it up a little bit and say what we're trying to do. So we got this, we had all these quite sophisticated ideas when I was a young scientist of how we could use Wolbachia. This Wolbachia was very effective at invading insect populations. So I really wanted to see whether we could harness that ability and use it like a Trojan horse. The story of the Trojan horse, where the soldiers pile into the horse and then foolishly gets dragged into the Troy, and the rest is history, as they say. And so would it be possible to put something inside Wolbachia and then have Wolbachia carry that cargo into a whole species of mosquito? That's what we were thinking about. And we had these really cool ideas of what the cargo could be and how it could interrupt dengue or malaria or and make GM Wolbachia. And it was all high tech and super cool. And fundamentally, we couldn't get any of it to work because we weren't able to genetically engineer the bacterial Wolbachia at all. It's still resistant to this day. No one's been able to find out how to put foreign genes into it. And then I was doing some work, reading about some of the amazing work Seymour Benzer was doing at Caltech, and one of his students discovered a different strain of Wolbachia that actually shortened the lifespan of the fruit fly Drosophila that it was living in. And they called this bacteria popcorn. And I was really intrigued when I saw this paper come out, that you had Wolbachia that was surviving in Drosophila, and it would over-replicate in the brains of these Drosophila and kill them early, would halve their natural lifespan. And when I read that paper, it was like ding, 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 ding. And the reason it was that is that when you know a little bit about mosquitoes and how they transmit viruses, it's only the old mosquitoes in the population that actually do the virus transmission. Why is that? And that is that so mosquito needs to take a blood meal to bite people to give it protein for its eggs. And this particular Aedes aegypti will take a little bite nearly every day if it can. And what it has to do is bite somebody who has the virus, dengue virus, and then that enters its body. And then the virus has to grow in its body before it can spit it back out in the future bite to another person. And that process can take up to 2 weeks. Well, 2 weeks for an adult mosquito is a very long time to live because, there are many things waiting out there to kill you, whether it be hot sun, whether it be a spider, whether it's a person slapping their arm in the middle of the night or whatever. There's many threats to the life of the mosquito that means it doesn't live very long. And so when Seymour Benzer published this paper about shortening the lifespan with Wolbachia, it was like, wow, we could get Wolbachia to invade the insect population. It would shorten the lifespan and reduce all the disease transmission. And we didn't need to do any genetic engineering. We didn't need to do anything. And so that was the idea that we actually pitched to the Gates Foundation. And we were able to put this strain of Wolbachia into the mosquito, which was a whole other complicated technical story. But then, what did we want to do? And then we wanted to test, in a sort of semi-natural setting, whether this popcorn Wolbachia in the mosquito could actually spread. And to do that, we didn't want to release it out into the field initially. We wanted to be a more controlled and have biocontainment on it so that we were working very safely and securely. And so we built in Australia these big outdoor cages that mimicked the natural urban environment for this mosquito. And we filled these cages with natural mosquitoes, and they were all living and breeding in there, like a mini house, all sorts of stuff going on in these huge outdoor cages. And in there, we wanted to test whether the Wolbachia could actually invade, could shorten the life cycle, the lifespan of the adult mosquitoes, but still invade. And we were able to show that, we thought it could, but then realized after that the fitness cost was so high, it was going to be very hard to get this strain of Wolbachia to actually work, if you like, to invade and maintain itself. And just at that point, something really unexpected happened to us. And after many years of failure, something really good happened, which was like, one of those days when the scientific result is amazing. And that was that we found that other strains of Wolbachia that we put into the mosquito as well, that didn't cause any life shortening, that hadn't been engineered or hadn't been changed in any way. But if they're just in the mosquito and we fed those mosquitoes with blood containing dengue virus, the dengue virus didn't grow in the mosquito. And we're like, what? And then we tried a whole bunch of other viruses, they didn't grow either. And then it was that point we realized that, wow, we didn't need to do any fancy tricks. We just needed a strain of Wolbachia and let it do its natural thing, and it would naturally give protection to the mosquitoes. And when the mosquitoes are protected, then they don't transmit the virus to people. And so this was like, like head spinning day in the laboratory when those results came in. And from that, that's now what the World Mosquito Program does. We take just this strains of Wolbachia that naturally prevent transmission, block the transmission of viruses like yellow fever, dengue, chikungunya, all of those viruses, Zika, and allow Wolbachia to do what it does naturally, and that is to spread into an insect population and then make those populations resistant to being able to transmit. But you started the question asking me about release kits, if you like, and part of this journey I've been on for my career, and the more I've worked in the field instead of the lab, I've become really appreciative of the need for simplicity to make things work. And so for two reasons. One, the work we do as a not-for-profit is targeting quite poor countries, many of them. And so there's not a lot of money out there to pay for treatment for these diseases. But also just to have an ease of deployment, and simplicity is what you want. You don't want things that are too difficult to do, particularly in difficult settings, where some of the places we work are very difficult. And so we drive for simplicity. And so what you mentioned, the Zanku kits, as we call them in Colombia, is just like a soda cup, where we take some eggs of the mosquito we grow in the lab that contains Wolbachia, and we drop it into this cup. We add some water and we add some fish food to it. And those mosquitoes grow, and then they fly out of that cup. We put it into the environment in a shady place, hang it from a tree, put it in someone's yard. The mosquitoes grow themselves and then release themselves. It's a very simple method for releasing mosquitoes into the environment. We set up a fairly, complex mapping system where we know where we're going to put these roughly, and we put them out into the environment, Wolbachia into the environment, and then it takes off. And what we see is very shortly after that, disease levels plummet close to zero.
Outstanding. Guys, as you were just, mentioning sort of this aha per the Wolbachia doing what it naturally does. You had one of the papers that there was a presentation you gave there. Actually, there's insect molecular biology for the Royal Entomology Society back in July of 2008, where you publish about sort of the interesting sort of defense in genes that mosquitoes have, and it just that one stood out to me just because I'm interested in sort of personally interested in sort of all the interesting ways that these species, whether they're insects or plants or what have you, have developed these defenses that potentially have other uses. But what did you find out in terms of obviously, the Wolbachia is doing some magic, whether it's upregulating this defensin or downregulating this. What did you find out on the thread? So were there any interesting findings that say, hey, maybe some of this defense in biochemistry or whatever you might call it may have other uses for us, potentially sort of bioprospecting related or otherwise? Because I hate to say, mosquito, they're all bad. Maybe they're 100% bad, but maybe there's some good stuff that is mixed in there that we can use potentially for other types of discovery work one day.
Yeah, so the work you're talking about is us trying to understand the mechanism of how Wolbachia did this, and part of that related to Wolbachia. We think two possible mechanisms that are acting simultaneously. One is that Wolbachia is sequestering key fatty acids like cholesterol that don't make that available for virus replication, and then the other is that Wolbachia is upregulating the innate immune system of the insect, so defensins, acropins, those other molecules get upregulated, which protects the insect from the virus infection establishing in the mosquito. But so that work has led to us trying to understand at a cellular level, what's going on, what's the mechanism here from Wolbachia, and possibly, there are there are features of the biology here that could be utilized, as you say. We could utilize molecules like defensins or other molecules to be used directly, if you like, or to put into engineered systems. A number of very smart scientists out there at the moment that are working out on very technically complex and elegant ways to sort of do what we're doing, but to do it in a more precise way without taking Wolbachia out completely, if you like, and just using the key molecules that Wolbachia produces. But I haven't gone down that path because, or we've stopped coming that because of this desire to be really simple with what we're doing. And not only that, to be doing something the public finds acceptable. And so there's still a lot of controversy around GM and people having GM mosquitoes released around them. Maybe in the future that will happen. Right at this moment, though, I feel like there's probably a lot of resistance to that idea, whether that makes sense or not from a technical or scientific point of view doesn't really matter. It's a political issue. And so, again, we've been very focused on practical. How do we get this thing to work in the field? And so we like simple, we like natural, we like non-manipulated systems because they're easier to explain and it helps people build trust. And ultimately, trust is what you need at the community level to do anything outside the lab.
Absolutely. Yeah. I was thinking more from the perspective of, whether they were antimicrobial or immunosuppressants or anticoagulants or all the other interesting biology that goes along with that mosquito, when it's sucking the blood and all that. But I get your point.
There is that. You're right. There's a lot of really cool stuff going on at a biochemical level, and that could be utilized for potential future interventions. And again, it will happen. I'm sure it will be interesting to see that unfold in the future. But I think what we're doing maybe is laying a simpler foundation, which will make it possible for more complex and technically elegant, if you like, approaches to come after us that are currently being developed in other labs. But for the moment, we're simple and robust and inexpensive and wanting to get out and have impact and for people, positive impact in people's lives.
Makes sense. So
instead of being in the lab, living the life in the lab.
Understood. Completely understand. Translation. I hear you. So obviously you're everywhere now, from Brazil to Indonesia. There was this major PR recently about the facility in Brazil and producing, I forget how many billions of bacteria to mosquitoes and
2 billion mosquitoes a year.
2 billion mosquitoes a year. Just talk a little bit about this. Any interesting stories, fun facts of what it takes to run a mosquito factory, any other interesting things regarding whether it's the Brazil initiative or other related translational sort of large scale spin-offs of your work.
So really early on, we've been really sort of taking community attitudes very seriously from the beginning. And because if for no other reason, if we put the ethical issue aside, if we just think about it in a selfish way, if you like, about wanting to get this technology out, if the community doesn't support it, it's going nowhere. And so we were very conscious about community support right from the beginning. And can you imagine what the first place would be where we went to this first town in Australia? We wanted to go to our own backyard first, and we wanted to release these mosquitoes. And it was a small farming community in far north of Australia. And actually, I don't know if you ever heard the story of the cane toad in Australia. But,
sure.
It was a biological control program in the 1920s that went bad and left all these cane toads rampaging over the landscape in Australia. Well, the place we chose to do our first experiment was the town that they released the cane toad in Australia. And there's like a kids park, cane toad world, and all of this sort of stuff in this town. So, this is a population that knew all about, something that could go wrong with what we're doing. And here we are, this group of scientists come in, and we come into town, we have a bunch of town hall meetings and meet with people individually and talk about our work. And we're saying to people, we're going to release, a whole bunch of mosquitoes here, and these mosquitoes going to bite you, and they're going to bite your children and your elderly parents. And by the way, we've put this strange bacteria into them, and we think it's going to be really great because it'll stop any dengue transmission in the community. But it's never been done before, and this is the first time we're going to do it. Are you with us on it? And it's a pretty big ask of the community to do that, and so. We put so much effort into the community engagement side of what we were doing. And I think I had lunch with everybody in that community just about. And we talked to everybody individually. It was a huge amount of work. But ultimately, the community valued the approach we took and I think the honesty of what we were doing and the transparency of what we're doing, and that we listened to their concerns. And they had concerns about things that we thought were scientifically implausible, but we did the experiments to show people with data that this probably wasn't the concern they thought it was. And so those early days around community acceptance were something else. It was quite challenging to get the communities to accept. But what we found is that if we did the work very authentically, we're very honest, we're very transparent, we're not trying to make profit out of it, we're not for profit, people got behind it. And what we've found in 11 countries now, wherever we go, we make the effort to do good community engagement, community supports what we're doing. And that's been, quite a profound thing, I think, for us in our journey, our own personal journey of translating lab results into the field. But what, to take it up a level, so we went from small farming communities in Northern Australia, where the communities are very trusting and very easy to access. But we took it from there to cities like Rio de Janeiro in Brazil. I don't know if you've been to Rio, but it's something else there from the point of view of, large, very poor areas, favela regions, as they're known, or slum areas that are virtually not under control. The government barely controls them. They're run by gangs, and they're super dangerous places. And so to take that idea, we're going to release all these mosquitoes, they're going to bite you, and then teleport it into a dysfunctional community where there's a very dangerous place where there are a lot of guns, there's a lot of security issues, a lot of drugs, there's a lot of violence, and it's very challenging for field workers to be there. And just the level of complexity would add it on to being able to do field work in that environment. But that's where the disease are, that's where we need to get into. And so, very interesting stories around the challenges of our field workers, and having people, mugged, having about people shot. Unfortunately, nobody killed. But, in Colombia, where we work in particular, it's quite dangerous for the field workers that we work in and work with us. And so, there's like many stories about all of the challenges of working in communities in very poor areas. And it's been an interesting journey. But across all of that, what we find is the communities are supportive, even in those areas of Brazil that I'm talking about, or Colombia. We find that once you take the time to talk to people in a very honest way and take their concerns very seriously and not be arrogant about it, and people value that you're trying to do something to improve their lives, and they tend to be quite supportive because there's not a lot of activities happening to support their lives, and so they end up being very grateful.
I think there's important learnings there for all of these I would call them moonshot programs, or however you want to refer to them,
Yeah.
but it's interesting. It's funny because when Eddie Holmes was on the show a couple of months ago, he specifically talked about. I mean, he's focusing on the the viral discovery side of the things from the Australian cane toad, but he did relate that story. So it's kind of interesting that
Yeah.
you had to sell into
Yeah.
that history. But no,
Yeah.
that's really
Yeah.
fascinating. Um,
Yeah.
Scott, what else is happening? Any other things coming up for 2023? I mean, I saw that next year you're going to be doing some stuff with the Gordon Research Conferences and Genetic Biocontrol Conference. Other things coming up for the rest of the year, other initiatives that we should know about, public-facing things, anything I missed, please let us
Yeah.
know.
Well, we'll be have people from the mosquito program at the annual tropical medicine meeting that'll be in October in Chicago. This year, it's a venue for people that work on these sort of diseases to present their work. Very large conference. We'll be there. For us, our journey came from the lab and was it possible, and then it was it then possible to actually do it in the field with support, and then it was the next step was what impact did it have,
Yeah.
and we did some very elaborate, very large, expensive clinical trials, particularly in Indonesia in the city of Yogyakarta, where we were able to show with like gold standard scientific evidence the actual positive impact. 86% reduction in hospitalization, 77% reduction in virus transmission. Those are like that's a similar level than what you get with condoms with HIV. We're talking about for a measured trial, very, very powerful impact. And so we've got all that done. What happens now? We've done all these pilots in 11 countries. We've got 40% of the world's population at risk of getting these diseases. That's a lot of people. And so the issue now is, can we take this knowledge and it's been tested in the field, shown to be acceptable, shown to be safe, shown to be effective? How do we get it out to 40% of the world's population that needs it? And so our focus is on scale up and what's the way to achieve scale up at the moment. And so we had public announcement from Brazil about how do we make millions and millions of mosquitoes. You think it'd be easy because they're a major pest, but when you have to grow them in a very precise way to get so many millions every week, week after week, it's quite tricky to do that manufacturing. And so we're very focused on how do we scale up manufacturing, how do we ensure quality, how do we scale community engagements so that we can deploy over large cities. And then what's the next pathway for release? Are we going to continue with these community cups that we hand out with communities, or to really scale up, do we need to think about releasing our helicopters? Or so we're interested in the technology side of distribution as well. So the next few years for us are all going to be about scale, demonstrating scale, and then transferring that knowledge to governments that need access to the technology for their own situations. And hopefully, our goal would be, at the moment, we've covered or protected during our research work around 11 million people now to date. And what we'd like to do in the next 5 years or so is to move the number up to around 500 million people protected. And that'll happen through partnership with governments, with other organizations, with other groups that have strong experience and ability to work in the field. And so the future for us in the near term will be all about scale.
I'm rooting you on, and I love the story. It's elegant. It's biomimicry. It relies on nature. It's self-sustaining. It's just it's such an impressive timeline, and the fact that, as you mentioned throughout the story, that you failed a lot, but nonetheless you came through and ultimately made this happen and affecting so many lives. So I just again wishing you the best with it all, Scott, as you continue to move it forward, scale it, and spread it to all corners of the earth. Really, really amazing work. Again, for everybody that is going to be listening to this particular episode of our show across the various podcast networks or watching on the YouTube channel. You've been listening to Dr. Scott O'Neill, professor and director of the World Mosquito Program, Monash University. Scott, I want to thank you again for taking the time out of your schedule to come talk to us for a little while. Obviously, thank you for everything you're doing for everyone around the world on this front, as we like to say on our show. Thanks for helping to create a better tomorrow via what you do. Really a great story.
Thanks for your interest and helping to spread the word of what we're doing. Appreciate it.
Absolutely.